Hi everyone, welcome back to my channel. This is MCQ practice paper 2. In my previous video, I have explained about transcription in eukaryotes and these 25 MCQ questions are based on it. So the link will be given in the description box. Kindly go and watch it. Question number 1. Transcription in case of eukaryotes takes place in option number 1, nucleus. Question number 2. If the sequence of bases in the coding strand of a double-stranded DNA is 5' GTTC GTC 3', the sequence of bases in its transcript will be option number 3, 5' GWCGA GUC 3'. This is because mRNA strand will be complementary to template strand of DNA, while coding strand has same sequence of RNA except thymine at the place of uracil. Question number 3. If a segment of an mRNA molecule has a sequence 5' GUACCGAUCG 3', which of the following could have been the template DNA molecule? And the option is 3. 3' CATGG CTAGC 5'. This is because adenine pairs with thymine and guanine pairs with cytosine. Question number 4. Heterogeneous RNA undergoes two additional processing, out of which in one of them as unusual nucleotide methylguanosine triphosphate is added to the 5' end of heterogeneous RNA. This is known as option number 1, capping. Question number 5. Genes are made up of option number 2, a portion of DNA. Question number 6. The central dogma is, and the option is number 1, direction of flow of information from DNA, DNA to RNA, RNA to proteins. Question number 7. The core enzyme which requires the factor for termination of RNA synthesis at some sites is known as option number 2, Rho factor, which is also called as Terminator factor. Question number 8. Which principle governs the principle of transcription? And the option is 3, that is complementarity. This is because RNA and DNA sequences are aligned anti-parallel to each other. The nucleotide bases at each position in the sequences will be complementary. Question number 9. Why both the strands of DNA are not copied during transcription? Because option number 4, all of the above. You know that if both the strands of DNA are copied, we are going to get two RNA sequences which will lead to double-stranded RNA, and this will not lead to translation. Question number 10. Which of the following is not a unit of transcription unit? And the option is 4, a operator. Question number 11. The direction of transcription is option number 1, always from 5' to 3' polarity. Question number 12. The strand of DNA which does not code for anything is referred to as option number 2, coding strand. Question number 13. Which of the following is true for coding strand? And the option is 4. Both B and C, that is both 2 and 3. Its polarity is 5' to 3'. It's a sense strand. Question number 14. Which of the following is true for promoter and terminator of a transcription unit? 1. Both flank the structural gene. True. 2. Promoter is upstream of the structural gene with respect to coding strand polarity. True. 3. Terminator is downstream of the structural gene with respect to coding strand polarity. True. So the option will be all of the above. Question number 15. Template and coding strands are defined by the presence of, and the option is promoter. Promoter because promoter site contains a sequence of nitrogenous bases that provides a site for the RNA polymerase to bind and initiate the process of transcription. It is located at the 5' end of the coding strand upstream, and with respect to the template strand, it lies towards the 3' end upstream of the structural gene. Thus, it marks the position of the template and coding strands in the transcription unit. Question number 16. In transcription unit, regulatory sequences are present. Option number 1, always at the upstream of the promoter, because promoter is the regulatory sequence. Question number 17. Which of the following is incorrect for a gene? And the option is 2, sequence of DNA which codes for a protein only. Because though the linear sequence of nucleotides in DNA contains the information for protein sequencing, proteins are not made directly from DNA. Question number 18. Which of the following is a false statement? And the option will be 1. Structural gene in transcription unit could be polycystonic in eukaryotes because polycystonic gene is only found in prokaryotes and lower organisms. Question number 19. Which of the following would be present in processed RNA? And the option is 1. Exons. Question number 20. Which of the following is incorrect? And the option is 4. mRNA, which is not matured RNA. Question number 21. How many RNA-dependent RNA polymerase are there for the transcription of all types of RNA in bacteria? That is prokaryotes. And the option is 2. Only one RNA polymerase will be present. Question number 22. In an eukaryotic cell, types of RNA polymerase present are not less than 3. There are 3 types of RNA polymerase, that is RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. Question number 23. Which of the following RNA polymerase is used for the transcription of hn-RNA? And the option is 1, that is RNA polymerase 2. Question number 24. Which one is or are correct? A. In prokaryotes, a single type of RNA polymerase can transcribe mRNA, tRNA and rRNA. B. In eukaryotes, RNA polymerase 1 transcribes rRNAs, whereas RNA polymerase 3 is responsible for the transcription of tRNA, 5S RNA and snRNA. C. RNA polymerase 2 transcribes hn-RNA in eukaryotes. D. Ribosomal large subunit has P and A sites. All these options are correct. So the option will be 2. A, B, C and D are correct. Question number 25. Which of the following RNA polymerase catalyzes the formation of transfer RNA or tRNA in eukaryotes? And the option is 4. RNA polymerase 3.